Morning. I was driving from Princeton to this direction to um, along the Route 33 this morning, and it was raining very hard. I was actually very pleased because a week ago I was driving in this direction too, driving toward Long Branch to run the New Jersey Marathon. So imagine running a marathon in this kind of weather. Okay, so I want to thank Melissa for giving a very nice introduction and also give a very good sort of a context about why we study metastasis of breast cancer. She mentioned that I'm the chair of the AACR Tumor Microenvironment Working Group. This is actually the largest working group of AACR. AACR is the largest international organization of all the cancer researchers. And this Tumor Microenvironment Working Group consists of over 8,000 researchers, 8,000 people like me globally, whose sole focus is looking at the tumor microenvironment, the kind of tissues that are surrounding the tumor. And why is this the largest group of researchers now? Because this is the most important direction in the field of cancer research and cancer treatment now. Some of the most recent breakthrough in cancer treatment, some of you already heard about immunotherapy, is essentially a study of tumor microenvironment, the immune cells that help us defeat cancer. So this is one of the major research area in my lab, and my goal today is to share you some of the work that we have done in the lab in the last 10, 15 years. And many of them are actually supported by funding by Komen. So you are part of it. Your support is part of the reason that we are able to conduct this type of research. So I don't need to go through this in detail because all of you in the audience know that breast cancer is the largest threat for women, not only in the United States, but globally as well. And it's actually even more so in many of the developing countries because in those countries, cancer are detected too late. And many of the patients are diagnosed with metastatic breast cancer at the time of diagnosis. So you can see many, more than 4 million new cases of breast cancer each year and more than 3 million cases of deaths from it. If you look at the world map in a different way, this lists the prominent cancer for women in each country in the world. And you can see it's almost entirely pink. It's mostly breast cancer in most countries. So when I started, I came to Princeton in 2004 after completing my training at Memorial Sloan Kettering in New York. And during my travel to visit the different universities, I was reading this Fortune magazine. And in this cover article, Clifton Leaf, who is a cancer survivor, discussed the issue about why we are investing a lot of research in cancer, but still many people die from it. Are we losing the war on cancer? So this is the article published in 2004, 15 years ago. And one of the major conclusions is that we sort of ignore the elephant in the room. Most patients die from metastasis. By the time when he was publishing this article, only 1% of national research funding is going to metastasis research. So there are two reasons. One is it's just too difficult to study. And second is a lot of the companies sort of losing the hope that when a patient has metastatic cancer, they feel it's a losing battle. So it's this mindset that sort of prevent us from getting into metastatic research in greater efforts. But that has been changing. I'm glad also Komen has been, as you can see, putting in a lot of effort. Actually, the large majority of the research effort funded by Komen are focusing on metastasis now, including especially people who already have metastatic disease. So when you think about why that is important, this is looking at the 5-year survival rate of people who have localized breast cancer versus people who have metastatic breast cancer in the 70s to the late 90s. And you can see that we are doing very well in treating people with early stage breast cancer. The 5-year survival rate is nearly 100%. But for people who are unfortunate to be diagnosed with metastatic breast cancer, the survival rate is 20%, 30, 40 years ago. And that is improving, but not in the kind of speed that we like, at least back until the late 90s. And we hope that we are changing that. So there are many thoughts that we think we could dramatically improve the survival rate of all breast cancer and reduce the mortality. One is, of course, early detection. You want to detect most patients in this stage. And for those people after surgery and adjuvant chemo, we hope the cancer never come back. And finally, for people who already have metastatic disease, the goal is to develop treatments that are not only able to maintain a good quality of life for extended period of time, but also hopefully cure the disease even after they already metastasized. So this actually has been the main goal of my lab, is to figure out how it happens, because as scientists, we always try to solve problems by figuring out what goes wrong. And once you know what goes wrong, you can find smarter ways to defeat that process. So there are two main questions that we have in mind. Is, one is this so-called crystal ball question. For a patient coming into hospital and being diagnosed maybe with early stage breast cancer, there's always this hanging threat in their mind that even after all the treatment that they go through, metastasis and recurrence might happen. So why there are some patients who are essentially cured after initial treatment, but why certain patients, even with a relatively small early stage primary tumor, will come down with metastasis a few years or even more than a decade after the initial diagnosis and treatment. So in the past, we can't really predict that. The pathologists could look at the tumor, but there's no certainty in predicting what is the chance of recurrence and how aggressive you should treat and monitor after the treatment. The second question is, if we can identify patients who have high-risk cancer or already have metastasis, can we just tell, do we just tell them the bad news? Or we can tell them that they have high-risk cancer, but we have drugs that will prevent them from metastasizing or from causing all the problems later. So in the field of molecular cancer genetics or genomics, the first question has been solved to some extent, and you probably heard of it. We can now profile patients' tumors using technologies, for example, like MemmaPrint. And they give out almost like a digital readout of the molecular profile of the tumor and give a unbiased sort of numerical number in terms of the relative risk of recurrence. And that help guide the treatment decision of, for example, adjuvant treatments for the patients. So this is one example of the initial study that paved the road toward this type of approach. It's a work published by a group in Netherlands, showing that if you look at the tumors, there are so-called poor prognosis tumors, bad tumors. They are likely to relapse or metastasize in the bottom half, versus the tumors that are so-called good prognosis tumor. They are treatable and likely to be cured. And look at the genes. Each column recommends a single gene. So you're looking at different tumors in the rows and columns in the genes. You can see they have very opposite gene expression signature, the green and red. So green means high, green means low, red means high. So you can see the almost opposite signature of those 70 genes in the bad versus not so bad tumors. So this gives us the technology to develop so-called MemmaPrint to do the digital analysis. But again, it only gives this prognosis, but for the patients who have bad tumor, we actually cannot tell them why they have an aggressive tumor and how do we stop that tumor from spreading. And so actually when I started my lab in Princeton 15 years ago, this has been a question that I've been constantly thinking about. How do we find the underlying reason behind aggressive tumors? So when you look back at finding the so-called driver genes that certain tumor behave badly, they often have a shared or common sort of phenomena. The location that harbor that gene is often altered in specific type of cancer. So you probably heard about this disease called chronic myelogenous leukemia. This is one kind of leukemia that often affect old people, something called CML. And a large fraction of them have this genetic alteration called Philadelphia chromosome, because it's first discovered at the University of Pennsylvania. And so that Philadelphia chromosome is basically something called translocation. We have two chromosomes, 9 and 22, they swap their DNA material. And this swapping accidentally create a monster oncogene that becomes dramatically active in those cells, the white blood cells, and drive their proliferation like crazy. So this is the so-called driver oncogene in CML caused by Philadelphia chromosome. And that led to the creation of the first molecular targeted drug called Gleevec that essentially turned this lethal disease into a complete chronic situation. You can just take the drug and you live basically until you die from something else. The same is true, another gene that you definitely should heard about, HER2, is discovered in patients. 20% of breast cancer patients have amplification of HER2 locus that drive the development of those aggressive HER2 positive tumors. So what we thought at the time is, can we find genomic alterations that associate with aggressive breast cancer versus not so aggressive breast cancer? And what it did is essentially give us a roadmap to find the location. So, this morning I came here because I have an address. If we have an address of this bad tumor gene, we can discover it. This is the goal. And what we did is, I'm not going to go through the mathematics here, so this is what we did in Princeton, is using an approach called computational biology to convert the data that we collect from patients, one kind of genetic information, into a precise map. So it's kind of machine learning, the way you understand it. And that convert this type of information into this type of information. And what we're looking for is these little peaks here. They are higher than normal. And that little peak give you an indication that a gene is probably hidden in that location. And in this case, in AQ22, that perhaps is driving those aggressive tumors. And this is a discovery that we made back in 2009. It's actually 10 years ago. It's just like Philadelphia chromosome was discovered in the 1960s, and the drug was developed 40 years later. But we are making the discovery in drug development faster, as you were seeing here. This is made in 2009. And you can see this is the location of AQ22 in thousands of patients that we look at in Europe and in America. They have the same kind of location. When you look at a tumor, these are the tumors collected at the Cancer Institute of New Jersey. And some of the patients have a lot of these orange dots. This is the location of that gene. You should only have 2 dots, one for your father, one for your mother. But this one have 5 or 6 or 7 because it's amplified, just like HER2 in HER2 positive breast cancer. So you can see this is looking at the survival of patients years after their diagnosis, 5 to 10 to 15 years. Initially, everyone are alive when they are diagnosed, but after that, over the years, some of the patients die. And you can see when they have high copy number of this gene, by 15 years later, 40% of them already die from breast cancer. But if they have low copy number of it, almost all of them are still alive. So this is a clear indication that this is doing something bad to cause trouble for those patients. So the work was published in 2009, and interestingly, that caught the attention of cartoonists of newspaper too, and they, of course, you can see in here, they try to associate this discovered gene with the corruption of politics in New Jersey, but that's sort of like, a separate ways of look at this stuff. Anyway, so that's the initial finding, but at the time when we found the gene, there's no idea about what does it do, why it caused the cancer to be behaving so badly. And second is, when you think about it, if it's a cancer-driving gene, if you try to drug it or target it, can you actually cause severe side effects in patients that make it intolerable for the patient to take the drug? So we also, at the same time, need to figure out what does it do in our normal body, because that will tell us what is safe to target it. So this is the work done by a graduate student at the time, and she just accepted a faculty position at University of Pennsylvania. So she's going to continue to work on this kind of problem, but in her own lab now. So, what Lilin did is create a genetically modified mice, and is using a clever approach to knock out this gene only in the mice. We have 20,000 genes in our body and 20,000 genes in mice, and we're only like 5% different between us and mice. Think about it. So this gene was knocked out in the mice, and so it doesn't have this gene. But interestingly, this mouse here have the gene knocked out, deleted but it still developed completely normally, which is actually good news. When you think about it, in the patients, if you try to eliminate this gene, it will still be fine. It's still alive and look fine. But these mice are not the regular mice. They are genetically engineered to express a very potent oncogene, just like HER2, in their breast. So by 3 months old, they already develop breast tumors. So this is what they normally look like. It looks like this mouse is fatter than the one here, but it's actually not because they are more fat. Those are the mammary glands of the mice. Mice have 5 pairs of mammary glands, and so when they develop tumors, they develop a lot of tumors. And they also metastasize to the lung, and eventually that's how they die from the mammary tumors in the mice. Curiously, this is the same kind of mouse that are predisposed to develop breast tumors. But if you eliminate this gene, you can see, first of all, they develop very little tumor, very little, and they grow very slowly. You know, this is how fast the so-called wild-type mice tumor grow, and this is how fast the knockout mice tumor grow. It's 10 times slower. And not only that, they don't metastasize to the lung, very little metastasis, and so the mice live much longer. So this is the most clean genetic evidence before we develop any kind of drugs. You can actually use genetics to show, first of all, this gene is not essential for the survival of animals or humans, hopefully. And second is, if you eliminate it, you cause dramatic reduction of tumors. So this is sort of like the ideal target for drug development. So this is another graduate student at the time, Andrea Blanco, and he's actually already a faculty member, assistant professor at UPenn, also at UPenn, for 2 years already. What he did when he was a graduate student in the lab was to find out how this protein works. And we have 20,000 different proteins in our body. How they work is they work together, they interact. You know, like I shake hands when I meet you this morning. That's how you communicate, right? So his work is to figure out what does this protein do, what kind of people it shake hands with, what kind of protein it shake hands with to carry out its job to cause problem in cancer. And what he did is go fishing. He used the material, this is the gene that we found, as a bait to go fishing. And when you go fishing, you pull out material in the bait, but you maybe pull out a fish that interact with it. And so he pulled out something in here, this little band in here. It turns out to be a protein called SND1. And this turned out to be the partner in crime that help material functions. And so I'm not going to go through this detail. This is the cartoon showing why this interaction is important. You probably heard about this term called cancer stem cell. This is the fountain of all the, origin of all the problems. Those are the tumor cells capable of seeding new tumors, including seeding new metastases. So cancer stem cell is the origin of all evil in cancer. But they are also under a lot of stress because it's all messed up in cancer cells. So they are under a lot of stress to die, basically. They survive because they have mechanisms to keep them alive under very stressful situations. And turns out that this interaction between MTDH and SND1 stabilize SND1 protein under stress. Otherwise, it gets degraded quickly. And this stabilization allows SND1 to support the survival of cancer stem cells. So this is why when you eliminate this protein or this interaction, the tumor will not form and the metastasis will not form. And they also become more sensitive to treatment. So we then use, again, sophisticated molecular biology to figure out how they interact, because we are now walking along the road toward drug development. And I'm going to show you how this is done. These two proteins interact because they shake hands to each other. And how they do that? The SND1 is in green in here. This is the green protein. On the surface of this protein, there are 2 pockets in cyan color here. There are little pockets of cavity here. And those pockets are inserted by the proteins that are sticking out in the surface of MTDH in yellow, and you have these 2 red residues, amino acids sticking out called tryptophan. Those are hydrophobic proteins that like to hide in cavities. They like to hide in cavities. So they found these cavities and they interact. This is how they shake hands to each other. So when you think about it, if you are a drug company chief scientific officer, this is a great possibility to develop a new drug. how do you block that interaction? Think about that as if you are going bowling. How do you go bowling? You use your fingers to stick into the holes of the bowling ball, and that's how you hold it, and you can swing it around. So if you try to make a drug to imagine this is MTDS protein and interact with SND1 in yellow here, and this is how they hold on to each other. How do you make a drug? You make a little thing that stick to the hole. So there's no way you can hold on to it. So this is exactly how you develop the drug. We use that. I'm going to go through the detail. We use this approach and the information from so-called structural biology. And we develop a compound that you can see in here in yellow. This compound is sticking into the hole in exact same way as the protein sticking the finger into the hole in red. So it's replacing that kind of interactions. And so the good news is we test this in the mouse models of breast cancer now, looking at the tumor growth and looking at the lung metastasis. And so you can see these are the mice treated with the compound we developed. You can see the tumor growth is significantly slowed down. But most importantly, the lung metastasis is reduced dramatically as well. So this is the first step. It's the first hit that we found. And the following work that we're going to do now is to try to develop better compounds that you could actually use in the patients, and we know it likely have very little side effect. And so this work is done by this postdoctoral fellow here, and he's a Komen fellow, so he's a Komen postdoctoral fellow. So this is the work is actually currently supported by Komen in the development of the drug. So this is one example about in a laboratory, you go from a initial clinical observation. The initial inspiration for our work is looking at the patients, why they behave differently. So it's a very broad question. By using the technology and tools that we have in the lab, it allows us to focus in a single gene. And then we use all kinds of different approaches. I already mentioned in this talk, the methods of molecular biology, genetics, biochemistry, structural biology, pharmacology, bioinformatics. These are all different disciplines of modern science, modern biology. But combine all of that, we can really lead from the basic science discovery to drug development. So I'm going to switch the topic a little bit because one of the main focus of my lab is studying this problem of bone metastasis. This affects large number of patients who have late-stage breast cancer. About 70% of breast cancer patients at late stage have stage 4, have some complication in the skeleton, bone pain, sometimes fracture, and conditions like hypocalcemia. So this is a major medical problem that we have to resolve. This is one example. A patient having bone metastasis, you can see the bone is being degraded, pelvis bone is being degraded, and that causes fracture and bone pain, many kind of painful complications. So it's actually not the tumors that are breaking down and eating out the bone. They are not able to do that. They are actually not specialized to do that kind of very difficult function. In our body, the only cell that can resolve or degrade the bone is this cell type called osteoclast. And we actually all have that because it's essential for our bone to function normally. So we have two kinds of cells. One is so-called osteoclast, one is so-called osteoblast, and they have opposite functions. So osteoclast resolve the old bone. This allows the osteoblast to lay a new layer of new bone. And this happens every day as we're sitting here. It's very important to maintain the strength of our bone tissue. Just like the driveway or the asphalt highway, you need to pave it. Once you have those potholes and everywhere, you need to excavate the old driveway and then put a new layer. This is essentially all those cells are doing. But they're doing that in a very coordinated way because you don't want to have osteoporosis or osteoporosis. That's the condition when these two are all balanced. So this is one situation in cancer that is all balanced. You see a lot of bone loss because the tumor educate and stimulate the osteoclast activity to a level that is hyperactive. You can see in here, this is the mouse bone metastasis. The bone in the left side, the tumor are growing. And in this interface between bone and tumor, those large giant cells are not tumors. Those are osteoclasts. So they are recruited and use essentially as false labels for the tumor to break down the bone. And that has been one of the major focus of the drug companies like Amgen to develop drugs to probably heard some of the drugs like bisphosphonates and denosumab. That's the antibody against Rankel. Those are all actually not targeting tumor per se. They are targeting osteoclasts to stop their function to break down the bone. So in bone tissue, these are not only cells that we are interested. There are also a lot of other cells that are involved in the normal bone physiology. There are this so-called, there are a lot of blood vessels. These are blood cells. Endothelial cells, those are blood vessel cells. And also many other like immune cells, fibroblasts, fat cells. Those are all resident cells in the bone marrow. So bone is actually an organ that generates most of our blood cells. So it's a very active hematopoietic system. So all of our blood stem cells reside in the bone marrow. And in the bone marrow, there are so-called niche, so-called stem cell niche that provide a specialized microenvironment for the stem cell to maintain their ability to renew and generate all the blood cells. Because our blood gets replaced every month, basically. The old cells get old, they die, and then you have to replace them with new blood cells. So there are so-called perivascular niche, the niche surrounding the blood vessels, or the so-called osteoblastic niche, the niche that are surrounding the bone-building osteoblasts. And those are essential for the hematopoietic stem cells to function. So it turns out that when the tumor disseminates from the breast to the bone, they actually try to take advantage of those niche environments that are already pre-existing in the bone marrow to their advantage for their seeding and for their growth. So this is, again, looking at a tumor in green metastasizing into the bone. And in the early stage of bone metastasis formation, you can see the tumors are always trying to hug in the blood vessels because the blood vessel seems to be showing a specific affinity. And when we have molecular study to show that in the bone, the reason why the tumor go to bone very often is in the blood vessel of the bone, there are a lot of those green staining. Those are staining for a molecule called E-selectin. E-selectin is expressed in our blood vessel cells to allow the leukocytes or the white blood cell to slow down and roll and extravasate. That's very important to fight infections when we have an injury in our blood vessel. In the bone, this E-selectin is very abundant. You can see a lot of these blood vessels have a lot of this green staining, but not in the liver or in the lung. And so it turns out that E-selectin in the blood vessel in the bone is essential for the initial seeding and formation of bone metastasis. In this mouse that we, again, use genetics to knock out E-selectin, you can see the formation of bone metastasis in the mice compared to the control mice, it's much less. So 10 times reduction of bone metastasis development. This is the work that was just published 2 weeks ago in this journal called Nature Cell Biology, and this is entirely supported by Komen. So why is this important? Again, this is a molecular interaction that you could actually try to disrupt. The tumor uses a receptor to bind to the E-selectin in the blood vessels, and this interaction actually requires the protein to be decorated by sugar. You're eating some sugar this morning. It's not only good food, but it's actually important for molecular interactions because the protein is also not just amino acids. They are modified with sugar in a particular way. So there are smart people in a company called Glycomimetics. This is a new startup company. Essentially what they do is make drugs that are sugar. They are so-called sugar mimetics. So it's called Glycomimetics. This is the company called. And so they invented this drug called, it's a sugar mimetics that mimic this sugar modification in the protein-protein interactions between E-selectin and its partner. So what we did in here is we treated mice with this sugar mimetic called GMI glycomimetic compound. And you can see significant reduction of bone metastasis development. You can see these are the mice treated with glycomimetic compound versus the mice that are not treated. You can see those red dots all means bone metastasis. So what this is telling us is that maybe for patients who have early stage breast cancer, All they need to do is take this sugar compound. Everyone likes to eat sugar. So it's safe because it's not a chemotherapy drug. It's basically a sugar mimetics. And that will help preventing those tumors to disseminate to the bone and form this initial bone metastasis. So this is something that the company is interested in going on in trials. But I'm going to end my talk with actually a work that is sponsored and also collaborate with one of the lead sponsor of the event today, Amgen. So this is a project that we started with Amgen about 15 years ago. And actually more like 10 years ago after the end of this project published in 2011. So this is a work done by a MD PhD student who did his medical part in Rutgers Robert Johnson Medical School and then the PhD in my lab at Princeton. And his discovery is one of these molecular crosstalks between the tumor and the microenvironment. Again, this is a tumor microenvironment study. So what happened is when the tumor metastasized to bone, they, as I mentioned, activate this so-called osteoclast in orange and brown here to degrade the bone. The bone is actually not like a piece of stone. The bone contains a lot of growth factors. So when it's been degraded, those growth factors are released. And one of the factors that is important is called TGF-beta, transforming growth factor beta. And it's a signaling molecule. So this signal was sent to the tumor cell from the bone to tumor cell and instruct the tumor cell to change the gene expression, alter its activity. And one of the genes that get turned on is called JAG1. So JAG1, again, is a signal molecule displayed in the surface of the cell. Again, this is another molecular handshake. So JAG1 interact with this receptor called Notch1, in the osteoblast and osteoclast. And it has two consequences. In the osteoblast produce growth factors like this that stimulate tumor growth. In the case of osteoclast, it stimulates osteoclast activity and recruit them to the bone surface to degrade more bone. So it's complicated, but the take-home message is they communicate, and there's a positive feedback of so-called vicious cycle. One signal leads to another, and it's just go on more and more and amplify, basically. So the way to think about therapeutic is, can you intercept this kind of communication? And one of the targets that we become very interested is JAG1, because Amgen, turns out, already is in the process of developing a neutralizing antibody to block JAG1. So it's perfect. We have a disease that is perfect to test this potential new compound that Amgen is developing. So we then test this in the mice first. So this is a experiment that hopefully I will walk you through. So in blue in here, this is our mice that develop bone metastasis without any treatment. And this is looking at how fast the bone metastasis grow in log scale. So each notch is tenfold increase. So tenfold increase log exponential growth of tumors. In yellow here are the mice treated with chemotherapy, paclitaxel, that you probably heard about. And just similar to as in the patients, initially the tumor responds to Taxol, so it slowed down. But later it becomes resistant. So you can see it accelerates at the end, and in the end it's almost like no effect. So this is very common that we see in the patients, especially in bone metastasis. They are very resistant to chemotherapy. They are not responding well. What we have is the agent that we developed with Amgen in red, and this is an agent that targets this crosstalk between tumor and the stroma cell through JAG1. So this is a JAG1 neutralizing antibody. So you can see it has some effect. It's about tenfold reduction. So it looks promising. But actually the most striking is if you combine this antibody with chemo, this is in green here, this is about 100-fold reduction. Again, this is in logarithmic scale, so it's all exponential. So instead of just tenfold reduction, combine both two drugs, you have a synergistic interaction. So you have more than 100-fold reduction. And you can clearly see that in the mice. This is mice without treatment with just the chemo treatment. And this is with the antibody treatment. The last group is with both treatment combined, and it's almost complete clean with tumor. So this is really striking. So at the time we thought that, well, maybe the chemo is killing the tumor and JAK1 is blocking its interaction with stroma. So that's how it works, and it really depends on JAK1 expressed in tumor to have this synergy. So we did another experiment in which is using a cell, cancer cell that do not express JAK1, the intended target here. And the expectation is you are not going to see the synergy in here. So here in red is the chemo treated one. So initially you have good response, but later it become also resistant, so it just accelerates. With antibody here in green, there's no response because the tumor do not express the target, the JAK1. Just like if you try to treat HER2 negative patients, you're not going to see response. But the surprising thing is that when you combine both treatment together, you still see nearly 100-fold reduction. So this is surprising to us because the target was not there. Why is it responding? And we scratched our head and we thought that maybe the phenomenon is because maybe the chemo is somehow inducing JAK1 in some cell, either the tumor cell or in the stroma cell, and is somehow protecting the tumor from chemo. And this is why when we combine chemo with JAK1 neutralizing antibody, you see a synergistic therapeutic benefit. This is kind of an unconventional thinking. So to make the long story short, that turned out to be the case. It's indeed the case. This is looking at the bone marrow of the mice and standing for this protein JAK1. So without chemo on the top, this is the mice without chemo. There is no JAK1 expressing in the bone tissues. But if you expose the mice to chemo, you can see the JAK1 start to be expressed. You can see those red dots. Those are standing for JAK1. So chemo is the inducing JAK1 in the mice. And so, again, this is complicated, so I'm going to walk through it. So the bottom line is we did studies to show that the chemo induced the bone cell to express JAK1, and that become a protective shelter for the tumor to protect them from chemotherapy. So this is counterintuitive, right? So when we treat patients with chemo, the goal is to eliminate tumor. And it's actually achieving that. That's why a lot of patients benefit from chemo. But we also unexpectedly creating a side effect that is actually, in the end, protecting tumors. The chemo induced JAK1 in the bone tissue cells, and that become a protected shelter, niche, for the tumor to die from chemotherapy. So we are in a dilemma, right? Because we try to use chemo, and because it's still effective. But in the situation of bone metastasis, it's not very effective because we are creating this response of bone tissue that is actually protecting the tumor. So this is what we figure out. And so again, this is showing you that these are the bone cells in red. If they are pre-treated with chemo, they become more protective of tumors in green here because they have this almost like allergic reaction to chemo, and in the end, actually protecting the tumors. So, but the good news is now we know how it happened, and we actually, more than that, we have an agent to block that side effects that is protecting tumors. So remember, JAK1 neutralizing antibody. The reason why it has this great synergy with chemo to have therapeutic benefit is because when you have chemo to try to kill cancer cell in the bone metastasis, it induces a response in the bone cell to produce JAK1 that end up protecting the tumor. But in this scenario, when we have JAK1 antibody, we eliminate that protection. So we treat the chemo and at the same time disarm this protection. So the tumor becomes sensitized to the chemotherapy. So this is what we hope for, is that for patients who already have bone metastasis, perhaps we can combine chemo with JAK1, and what we hope to see is what we hope to see as in the mice. Not just a 10-fold reduction, but 100-fold or more than that, to potentially a complete response to this kind of treatment. Moreover, we could also think about how do we improve our adjuvant chemotherapy for patients who have early-stage breast cancer, but based on mammoth print, predict that they are likely to have recurrence. So they are in high risk. And we need to have more effective adjuvant chemo to wipe their bone marrow clean to prevent later recurrence, 5, 10, 20 years later of bone metastasis. And this is one approach, is to, instead of just treat them with chemo, you should treat them with chemo combined with JAK1 neutralizing antibody, because in that scenario, it will make the tumor much more sensitive to the adjuvant chemotherapy. And this is what we try actually in the mice. So this is mimicking the situation in the patients. You make a primary tumor in the mammary gland of the mice, and then usually in a few weeks, 4 weeks later, they will develop bone metastasis. But we now apply treatments, either no treatment or the JAK1 antibody or chemo or combined treatment. And this is looking at the incidence of bone metastasis. Without any treatment, there's about 55% of bone metastasis incidence. With single treatment, and that reduces about 10 to 20%. But with the combined treatment, only about 10% of mice develop bone metastasis later. And the number of lesions, the bone lesion area is also dramatically decreased. So this is what we hope Amgen will continue to do in the clinical development, is to, maybe apply the treatment to the late stage patients who have bone metastasis to see if it's much more effective treatment than maybe the current standard of treatments for the patients. And second is, once that is approved, maybe extend that to a adjuvant setting to prevent, because that's the best window, right? Before any kind of clinical sign of metastasis occur, that's the great window to have a treatment that hopefully will just wipe the body clean and never have a recurrence. So I'm going to end my talk by just thank the people in my lab. You can see these are the young people in the lab who actually did a work that I show you today. And the good news is that many of them are now professors on their own. So more than 10 of them are professors in the US. 3 of them are in UPenn, and 1 is in Notre Dame, some of them in Europe, some of them in China. But they all are still doing breast cancer research. So we are proliferating too, very fast too. and the hope is that the combined brain power of all these young and talented and very dedicated people eventually will lead to the defeat of breast cancer in an actually foreseeable future. That's my hope. Thank you very much.